Welcome to Module 5. This module provides a high-level overview of the GRAND clinical trial, including its goals, study hypothesis, trial design, and schedule of visits. The study is designed to generate all of the data that the FDA will need to evaluate the system's safety and effectiveness. Many of the features of the clinical trial have already been worked out with the FDA in preliminary discussions, including the choice of outcome measures, the study hypothesis, and the final sample size, which is expected to be 30 participants. By completion of this learning module, participants will understand the structure and processes of the GRAND pivotal trial, list the inclusion and exclusion criteria for participant candidacy, describe the primary and secondary efficacy endpoints for the trial, identify the primary safety endpoint, and understand the participant timeline for study participation. As a note, we will be using acronyms from time to time during this learning module. Please refer to our supplemental glossary accompanying your training materials. The GRAND trial will implement an implanted network neuroprosthesis to restore grasp and reach in 30 participants with cervical spinal cord injury. Primary effectiveness of the network neuroprosthesis, or NNP, will be measured using the grasp and release test, administered at 3 months post-implantation, where comparisons will be made in performance with the NNP on and off. Therefore, participants will act as their own controls. While the key timeframe for assessing this outcome is at 3 months post-surgery, the GRT will also be administered at 6 months and 1 year post-implantation to confirm that the 3-month results demonstrate consistency of the system. While the primary outcome measure is the GRT, we will also include a few secondary outcome measures to assess effectiveness. We'll talk more about all of these measures in Module 8, which discusses primary and secondary outcome measures in detail. Safety of the network neuroprosthesis will be assessed by detailed recordings of adverse events. Adverse events experienced by 6 months post-implantation will be reported to the FDA for initial submission for approval. However, the GRAND trial will monitor and report adverse events for the entirety of trial participation. Therefore, continued data collection at 6 and 12 months post-implantation is critical for success of the GRAND trial. The following are inclusion criteria for the GRAND study. These criteria will be reviewed in greater detail in Module 6, which focuses on participant recruitment and screening. At face value, the ideal GRAND study participant is age 16 or older, has a cervical level spinal cord injury with retained shoulder function, but loss of function in the forearm and hand. They are neurologically stable and have intact innervation of the paralyzed muscles in their forearm and hand. They have a desire for greater independence and are able to understand and provide informed consent. Unfortunately, not everyone with a cervical spinal cord injury is a candidate for the NNP. The following are conditions that exclude persons with SCI from candidacy. Again, we will review these exclusion criteria in greater detail in Module 6. As mentioned, the primary efficacy outcome measure is the GRT, measured at 3 months, comparing performance with the NNP on and off. Improvement in the ability to grasp and release at least one additional object as compared to performance without the NNP will be classified as a success. We recognize that there are many criteria for success as a result of the NNP. So, we have included additional or secondary measures of efficacy, including grasp strength, which compares forces generated from the hand with the NNP on versus off, the Canadian Occupational Performance Measure, which measures changes in performance and satisfaction of goals identified by the study participants, the Activities of Daily Living Abilities Test, which uses a scale to quantify level of independence in performing activities identified by the COPM, and the Spinal Cord Independence Measure 3 Self-Care Scale, which assesses ability to perform self-care activities. Additionally, the NNP has the capacity to log or record device usage, which serves as a measure of device utility. Finally, participant satisfaction will be rated using the Participant Satisfaction Survey, which measures satisfaction across all aspects of device performance. Again, all of these measures will be reviewed in greater detail in Module 8. Safety of the NNP system will be assessed by monitoring adverse events. The safety profile for the system will include an analysis of the surgical complications, as well as adverse events reported throughout the study. The safety profile will characterize adverse events at 3, 6, and 12 months post-implantation. Module 9 reviews the procedures for adverse event reporting in greater detail, including definitions for adverse event severity, relatedness, and expectedness. Both our Data Safety Monitoring Committee and the FDA review these reports every 6 months, and Module 9 will go into detail on those reporting requirements. To gauge success of the GRAND trial, post-surgical outcomes with the neuroprosthesis on will be averaged across the participants and compared to post-surgical outcomes with the neuroprosthesis off. Our null hypothesis is that there is no difference between outcomes with the NNP on compared to off. However, based on the GRT, our primary outcome measure, we hypothesize that across the study participants, the number of GRT objects that can be moved with the neuroprosthesis on at 3 months is significantly greater than the number of GRT objects that can be moved with the neuroprosthesis off at 3 months. Additionally, a clinically meaningful difference for an individual study participant is the completion of 1 additional object on the GRT. The remainder of this learning module provides a broad overview of the schedule of study visits. It identifies what activities are performed by the clinical trial site and what activities are performed by the sponsor. As this is a broad overview of the study, detailed information about some aspects of the study will be included in additional learning modules. Prior to enrollment in the GRAND trial, participants who are candidates for voluntary reconstructive procedures, such as tendon transfers or nerve transfers, are referred to the appropriate clinical reconstructive surgery program. After tendon transfer or nerve transfer rehabilitation is complete, at approximately 6 to 12 months post-surgery, these participants are eligible for GRAND screening and enrollment. In other words, voluntary tendon or nerve transfer procedures will not be performed in conjunction with implantation of the NNP system. This is the timeline of visits and activities for the GRAND trial. We'll break down the timeline and look at each phase independently. Screening, enrollment, and qualification activities will result in the knowledge about whether or not someone is a candidate for the NNP at a given point in time. While NNP surgical implantation will take place in Cleveland, these activities will be conducted by the clinical trial site. Key aspects of assessment include obtaining a detailed history and physical, conducting an expedited INSCI, manual muscle testing and stimulated manual muscle testing of the upper limb, and additional tests to rule out any conditions that preclude participation. Once collected, screening data is transmitted to Cleveland, and participant candidacy is confirmed during a joint video meeting involving the Cleveland team and the clinical trial site. After participant candidacy is confirmed, the process of surgical planning is initiated between the sponsor and the site. The study PI and key team members will lead this effort in tandem with the site PIs and study team. This is meant to be an iterative learning process to help get sites up to speed with independent surgical planning. The surgical planning process is reviewed in greater detail in Module 11. Finally, it's important to note that there may be a variable amount of time between screening and surgical implantation due to different study-related factors and technology availability. In some cases, certain aspects of testing may need to be repeated prior to implantation, and it's important to make sure that the prospective participant is aware of this possibility. Once a surgery date is identified, study visit 1 should be scheduled for conducting baseline assessments and pre-surgical testing to clear for surgery. The baseline assessments, which include the primary and secondary outcome measures listed here, are described in greater detail in Module 8. The baseline assessment visit and associated activities take place at the clinical trial site within 30 days of the scheduled surgery date in Cleveland. We recommend testing to be completed by 3 weeks prior to surgery to address any minor problems that can be addressed without canceling surgery. For example, if a UTI is detected as part of pre-surgical testing, it may be treatable with antibiotics without having to postpone the procedure. For all data collection activities, timely transmission of data to Cleveland is paramount. Study Visit 2. The NNP implantation surgery, postoperative management, and device programming activities take place in Cleveland during Study Visit 2. The participant should plan to arrive in Cleveland 2 days prior to surgery. Complimentary lodging is available for the participant and family at the Zubizarreta House, a fully accessible bed and breakfast facility adjacent to Metro Health's Center for Rehabilitation. The participant will meet with the Cleveland team the day before surgery to review muscle excitability and address any questions related to the surgery or other study activities. The NNP implantation surgery will take approximately 6 to 8 hours. The participant will likely be in the hospital 1 to 2 nights following the procedure, depending on how well surgery is tolerated. The participant is discharged from the hospital to the Zubizarreta House until the first post-operative visit, which takes place approximately 1 week after surgery. During this visit, the cast will be removed, surgical wounds will be checked, and a new cast will be fabricated. The participant will wear the cast for 3 weeks following surgery. The participant and family may elect to stay at the Zubizarreta House through Study Visit 3, commencing at 3 weeks post-surgery. Study Visit 3. Visit 3 takes place at 3 weeks post-surgery in Cleveland. During this visit, the cast is removed and the NNP is activated for the first time since implantation. The goals of this visit are to assess the responses of the stimulating and recording electrodes, a process which we call electrode profiling, and program an exercise regimen to strengthen the implanted muscles. More details about this process are provided in Module 13. The length of this visit will be approximately 2 to 3 days. The participant will leave this visit with an exercise regimen using the NNP for home use. This exercise regimen will build strength and endurance of the implanted muscles in preparation for functional mode programming, which will take place at Study Visit 4, occurring at 8 weeks post-surgery. Arrangements will be made between the sponsor and the clinical trial site to participate in as many aspects of this process as possible, either remotely via Zoom or in person as allowed. Study Visit 4. The participant returns to Cleveland at approximately 8 weeks post-surgery, after about 5 weeks of exercise using the implanted system at home. The purpose of this visit is to program grasp modes to be used for functional activities and to establish myoelectric control parameters that are effective for operating the NNP. The participant will be trained in all aspects of system use. As with prior visits, Cleveland will facilitate remote or in-person participation for key aspects of the visit, as desired by the site. This visit will last approximately 1 week. At the end of the visit, a summary meeting will take place with the sponsor, participant, and trial site to review the functional modes, control mechanisms, system operation, use of the app, and to address any concerns. This meeting and completion of Study Visit 4 serves to pass the baton to the clinical trial site for data collection during Visit 5. Study Visit 5. Study Visit 5 is a critical visit for data collection and takes place at 12 to 13 weeks post-implantation. All study activities will take place at the clinical trial site. The goal of this visit is to administer the primary and secondary outcome measures. Study Visit 6. Study Visit 6, which takes place at 6 months post-implantation, is also a critical visit for data collection. All study activities will again take place at the clinical trial site. The goal of this visit is to repeat administration of the primary and secondary outcome measures. Also at this visit, participants will be asked to complete a satisfaction survey, about their NNP experience. Study Visit 7. Study Visit 7 takes place 1 year post-implantation and is the final visit for primary and secondary outcomes data collection. All study activities will take place at the clinical trial site. It's important to note that the sponsor and clinical trial site will be monitoring adverse events throughout the study, as detailed in Module 9. While this visit marks formal completion of functional data collection, participants are encouraged to contact the site and study sponsor to report any adverse events. We hope you have a good idea about the structure and processes of the GRAND pivotal clinical trial, including understanding who are candidates for the NNP, how we will measure efficacy, and the general timeline for participation. Upcoming modules will provide greater detail about key aspects of the study. Congratulations, you have now completed Module 5. Next, we will swim through a deep dive on participant recruitment and screening.